This is a production of Cornell University.
Yeah, good afternoon to everyone. Thank you for your interest in this session. So yes, I'm Diana Obregon. I'm a PhD student in the Entomology department at Cornell. And today I want to show you the results of an experiment that we did in 2019, trying to understand how different thiamethoxam applications in squash crops can impact pest control, but at the same time, pollinators. So we know that growers that cultivate crops that are highly dependent on pollinators such as apple or passion fruits or blueberries, they are facing a constant dilemma because they need to find the best practices to control the pests in the crop, but at the same time they need to protect pollinators in order to get the maximum yield. So they are facing all the time they are looking all the time for these new practices that can help them. And this is the case for cucurbits, for squash, for pumpkins, for watermelons, because these crops are entirely dependent on animal pollination to produce fruits. And this is because they have separate female and male flowers, and the pollen is very heavy and sticky, so they really require the work of bees to do the pollination in order to produce fruits. And in this region, the main pollinators are the squash bees, the bumblebees, and the honeybees. And in particular, I want to highlight the squash bees because these are specialist bees. They feed only on cucurbits, and they nest in the ground between the crop field. So anything that we do in the crop might impact this bees. And I wanted to show you just how the nest entrance looks like. So you can see the holes that they made and all the soil around. And if we have the opportunity to look at the profile of the nest, you can see that they build a tunnel with different cavities where they storage the pollen that they collected from the crop, and then they lay eggs, and the larvae will feed from the pollen. And from our data in that experiment, we reinforce the idea that bees are important for these crops. So you can see in this graph that as the number of bee visits increase, there's an increase in fruit number, but also an increase in fruit weight. So really, these bees are contributing to the crop production of these species. But on the other hand, we know that we have pests too. So the main pest in this system is the striped cucumber beetle. And we know also from like previous studies and from our study that when there's an increased defoliation caused by this beetle, there's a decrease in fruit number per plant. So these pests can really diminish the yield, but also we know that they can be vector of bacterial pathogens. So what growers commonly do is that they apply neonicotinoids at the beginning of the crop as a prophylactic measure, and then they usually spray like 2 or 3 times more with pyrethroids. But neonicotinoids in particular, there has been a lot of attention because of their effect on bees. We know that these are systemic insecticides, and in cucurbits, the main molecule that is applied is thiamethoxam. Yeah? Oh, I thought you were calling me. So the main molecule that growers apply in this system is thiamethoxam. And this molecule has been registered for in soil treatments and foliar sprays for several years. And since 2009 has been also registered as the insecticidal component of seed treatments by the name of Cruiser. And but we know that because these molecules are systematic, they can move throughout the tissues of the plant, and they can move, for example, from the roots to the flowers or from the leaves to the flowers, even reaching the pollen and the nectar, so the bees can be exposed to them. And this is unfortunate because these molecules are highly toxic for the bees. So in our experiment, we wanted to test how this different prophylactic application of thiamethoxam impact pest control, bee visitation, yield, and the pesticide residues in the flowers. And our goal was to try to find if any of those different application methods can provide a good level of pest control, but at the same time reduce the pesticide risk for the bees. And for that, we use these three products. We use Platinum by Syngenta, that is designed to be used as an in-forward treatment, Actara as a foliar spray, and Cruiser as a seed treatment. And you can see already that the different treatments provide different amounts of active ingredients. So for example, for Inforo, you get at the end, like doing all the calculations, you get like 0.028 grams per plant in comparison to foliar spray that you get half of it. And for seed treatment, it's much less than that. And for our experiment, we also wanted to test two different plant varieties to have different pest pressure scenarios. So we use golden zucchini, that is a variety that is highly preferred by the beetle. And we also use Success PM, that is a variety that is less preferred by the beetle based on previous studies. So this is how our experiment looked like in the field. So we did it in Freeville, New York, and we had these 4 treatments, including a control with no insecticides, the 2 varieties, and per each combination, we have 7 replicates for a total of 56 plots. And every plot contained 24 plants, and you can see the distribution more or less that we had. And we measure in every plot, we measure the percentage of defoliation that was caused by the striped cucumber beetle, the visitation of the bees, the number of fruits, the fruit weight, and also during the flowering, we collected samples of nectar and pollen at 2 time points. So at the beginning of the flowering in early June, and then 2 months later in August, to have 2 moments and to see if like the pesticide decrease with time, the pesticide residues. And to know if the pesticides that we found in the flowers can represent a hazard for the bees, we measured these, we use this calculation called the hazard quotient. And basically it's a relation between the mean concentrations of the residues that we found in nectar and pollen multiplied by the exposure, so how much nectar or pollen the bees consumes or get into contact with it, and this divided by the oral or contact LD50. So LD50, it's the lethal doses at which 50% of the population that you are like testing will die. So if we do these calculations and we get hazard quotients that are equal or above 1, that indicates that the levels of exposure to the pesticide can cause 50 or more % of mortality, and that represents a lethal hazard for the bee. So we are going to calculate this hazard quotient for squash bees that we learned before, that is the most sensitive species in the system. Okay, so these are the results. First, we look at the defoliation. So you can see that we have in the x-axis all the different pesticide treatments, and in the y-axis the percentage of defoliation caused by the striped cucumber beetle. The only differences was the in-forward treatment in relation to the others. So in-forward had the least defoliation in comparison to seed treatments, foliar sprayed, and no insecticides. And as you see, it's surprising that the other 2 insecticide treatments were not different from the control treatment. For the plant varieties, as expected, we found that golden zucchini had more defoliation than Success PM, and we expected that before. Now for the yield, so in the left we have the fruit number, and we can see again that Inforo is the treatment that is different. So it produced more fruit numbers than the other treatments, and there was no difference between seed treatment, foliar sprayed, or no insecticide. And we found basically the same for the fruit weight. So Inforo had the highest fruit weight in comparison to the rest, and foliar spray was even lower than the control, than the no insecticides. In relation to the bee visits, we saw that the treatments with the highest number of bee visits were the seed treatment and the Inforo treatment, and in particular for the Success PM variety, and this was because they had less defoliation. They also had more flowers, so that resulted in more visits per plot. And these are the residues, the thiamethoxam residues that we found in the flowers. So there's a lot in this table, but in the top you can see the 4 treatments. Early means that results from the early flowering, so in early June, and then late, that was in August. And then you can see the results for nectar and pollen, and then the frequency of quantification, that's the percentage of samples that we found with some level of pesticides, and then the range, that's the concentration of the pesticide that we found. And as you see, for the in-forward treatment is actually, again, the treatment that is very different. So you see that there's the higher frequencies and also higher concentration of the pesticides. We also measure clothianidin, that is the main metabolite of thiamethoxam. So that when thiamethoxam is absorbed by the plants, it can degrade into clothianidin, so we need to also check this type of molecules. So in this case, we found residues of clothianidin just for this treatment. So again, it's like the treatment that was different from the rest. And you can see here just the treatments, and in the y-axis the concentration, and again the highest concentration was found in the in-forward treatment. But how lethal are those concentrations, right? Because we can see like it ranges between like 0 point something to 40 something, but how bad are they? So when we calculate the HQ, the hazard quotient that we talked before, there was just one condition where the levels were above 1. So that means that for that condition, there's a lethal risk for the bees. So in this case, you can see I highlighted with a red line. The condition is when you use in-furrow treatments and you sample flowers at the beginning of the flowering season. At that moment, this treatment represents a lethal hazard for squash bees in particular. And this is pretty similar to what was found in Ontario, Canada by Susan Chan. She did an experiment in hoop houses where she had bees nesting in different treatments. You can see in the right the different treatments that she tested. And she found that the number of nests that the squash bees could establish in these hoop houses were dramatically reduced when there was application of imidacloprid to the soil. So this is a similar neonicotinoid compared to thiamethoxam. But we can see that for seed treatment was not that bad, or for foliar sprayed with this insecticide. So like these applications to the soil are very bad for the bees. So in summary, what we found that in-furrow applications resulted in the less defoliation, in the highest fruit number and the highest fruit weight, but also at the same time, it resulted in the highest pesticide residues with the potential to be a lethal hazard for squash bees, in particular in the early flowering. And surprisingly, we did not find difference between the seed treatment and the foliar sprays in comparison to the control, which is worrying because we know that the seed treatments are the most common the most common application method. And in relation to the varieties, we found that Successpian had the least defoliation. So it's an interesting to see that there's a variety that it's less susceptible to the striped cucumber beetle, but unfortunately we didn't see differences in yield, significant differences in yield. So what is our study saying or showing? Is actually that the commercial applications that we use currently of thiamethoxam, they don't really provide a sustainable solution for squash growers, because for one hand, in-furrow was really good to control the pest, to produce a good yield, but it's also a risk for the bees in the short and the long term. And on the other side, seed treatments were not as good as we expected. But what can we do in the meantime? We really need to keep looking for solutions, right, that the growers can implement in their fields. But in the meantime, we suggest that that growers need to avoid the neonicotinoid use or limit it to seed treatments. They should implement and use pest thresholds. If they need to apply it, they could try to apply it after the flowers have closed. So this is usually for squash crops in the afternoon. We also suggest that they should avoid tan mixing of insecticides and fungicides because that increased toxicity, and I'm sure Maria will expand on this information. And also for specifically for cucurbits, you should avoid to do pesticide applications over the nesting aggregations. So when we saw the nest, they're usually aggregated, so you should avoid to like destroy or contaminate their nest. And something that you can also do is leave a margin of bare ground to ensure that there's some space for the squash ground nesting bees. And with that, I want to thank you, everyone, the sponsors. So this work was funded by USDA, and I am funded with a Fulbright scholarship from Columbia. And I'm glad to hear or any questions or comments.
Dan has a question here. Could you show the slide that has the table of HQ values you found? And you're going back to that.
Do you have a particular question about that then?
Yeah, I'm just wondering, you show the HQ is only above 1 for female contact oral in the nectar. Is that correct?
It's only above 1 for when you have from when you did in forward treatments, but just for the samples of flowers that we took at the beginning of the flowering season. So like the first week of June, we collected these flowers and we found levels that are lethal for squash bees. And the calculation is done for the whole life cycle. So we take into account the amount of the pollen that the larvae eats, the amount of pollen that the adult are in contact with, or also the amount of nectar that they can ingest. And this is based on previous studies of the species, of these bee species.
Okay, and were you seeing a difference in, I thought an earlier slide showed that the bees were still coming to the flowers later in the season. Is that correct or no? Even with the
they're coming all the time. They actually prefer plants that were treated with in-furrow or seed treatment just because they have more flowers and they have less defoliation. So bees are actually attracted to these healthier plants that were treated with the highest concentration of the pesticide.
I guess my last question, I'm just kind of wondering, the hazard quotient is above 1, but the flowers are still being visited and the yield is still being increased.
Yes. So the thing is that it's not, the bees will go to the flowers. They feel attracted to those like beautiful big flowers, and they will collect this nectar and pollen that is contaminated and can be lethal for them. But they won't die in the flower. They will die somewhere else, in the nest or somewhere else. So the actual decline in bee population, we will see it through time. So if we have the opportunity to measure a field that is treated with in-furrow applications through time, we will see a decline in bees, but we won't see it right away.
Okay, thank you.
All right, hi there. I am glad to be here presenting pesticide risk to bees today. I'll be discussing what we know and what we need to know better. And I wanted to start this out by showing you where exposure really occurs for bees, and it's on the pollen. And so I want to show you various bee species and how their babies survive. So right here we have an egg that hasn't hatched, but as soon as it hatches, it'll gobble up all that pollen. Adult bees eat very little pollen. They mostly collect it for their young. So this is an osmia. We have a megachilid here in nests and leaves. We have the squash bee that Diana was just talking about. And then this is a Coletes. And so they all eat it they prep their meal very differently. And then here in the middle, what I'm going to be talking about the most is the honeybee, and they feed, daily they feed their larvae, their pollen bread. So I just want you to keep that in mind as I go through this talk. And let's see, I can't change the slide. Here. Okay, so since 2006, we've seen 40 to 68% of colony bee loss per year. And this last year, we lost, out of 100,000 hives that we have here in New York, we lost 56,000. So that's 56% of our hives we're losing. And at the same time, we know that we have also seen a decline in our native bee populations. So from previous and current research, we know that at least 53 of those native bee species are experiencing some form of range contraction or population declines. And in a couple cases, we haven't seen these bees in about 10 to 15 years. So why is this happening? Well, there are a multitude of factors presented here. I'm going to focus on pesticides because this is, I would like to focus on this because this is the one factor that we have some kind of control over, either with our voting power or our research power and our education power. So I'm going to talk to you about what we know about pesticides and the impacts they're having on bees. So here's a little map of where I'll go in this talk. First, we're going to talk about about defining pesticide risk. So what do scientists and regulators mean when they talk about risk? And Diana really went over this quite well, but I will just refresh you in reference to where we're going in this talk. Then you'll see some examples of where there is risk and where there is not risk. And then we'll move on to what we need to know better. And we know a little bit, but what do we need to know better? So where's exposure coming from and what's going on? Why are we so focused on fungicides all of a sudden? Okay, so for defining risk, if I or someone else talk about risk, what does that actually mean? This is a simple means of understanding it. Risk is two things. It's exposure, so how much of something are you exposed to? And then toxicity, how bad is that thing for you if you ingest it or you contact it? Okay, so we typically use a metric called the hazard quotient. There's other metrics, but regulatory agencies, researchers, and the pesticide industry typically use hazard quotient. So here, I'll review this quickly, but Diana really did get a good review of it, but we do it in a little different way. Okay, so for a typical fungicide, during pollination, I'm showing you this example of the bee getting exposed to 1,000 parts per billion of this fungicide. So that's how much fungicide the bees are exposed to. And from other studies, we know that this fungicide is that if the bee comes into contact with 100 micrograms of this fungicide, at least 50% of the bees that come into contact with this amount of the chemical will die, or more, 50% or more. So this is known, that's why this is known as the LD50, that's interchangeable with toxicity in this case. So if we combine those two things, we have a hazard quotient of 10. While 0.1% of an LD50 is really low, if we could take that same amount, the same measure, this 1,000 parts per billion, and we can take a really toxic chemical like imidacloprid, which has an LD50 of 0.004. And with this, if we do 1,000 parts per billion, we can end up with 250,000 hazard quotient. So this is 2,500 over the LD50. So this is the same amount of chemical, but now you have much more risk because that chemical is so much more toxic. So this is an extreme, right? Like rarely do we see this in the landscape. What is really common to see in the landscape is something more like 40 parts per billion of imidacloprid. So you multiply that and we get 10,000, it has a quotient of 10,000. Okay, so that's a lot less than 250,000. But this is still like, this 10,000 is 100% of an LD50. So, you know, a population of bees getting exposed to 40 parts per billion of imidacloprid, they run the risk of about 50% of that population dying. Okay, so all of this is just to say what is going on when we're measuring risk, combination of exposure and toxicity. So that's what risk is. Let's talk about when there's risk and when there is not risk. Okay, so in the pesticide analysis lab that Scott McArt helps run, they have ran this is based on around 4,000 pollen samples that have come through that lab. And what they find that during day-to-day beekeeping, the pollen samples that are just coming in throughout the year, there's barely any risk from pesticides. However, once bees, once the spring starts and the bees start visiting flowering crops, that risk starts to escalate extremely. So it's all happening during flowering, and not surprising because that's when the bees are coming in contact with it. But just to point that out. And for an example of this, I'll show you a research project in New York apple pollination with honeybees. This is 30 orchards that we've been on and off visiting for the last decade. And the lab, researchers went out and multiple times a week, they sampled pollen provisions from each orchard. And then they took those pollen provisions and one by one, put them through the pollen analysis machine. And they popped out a list of pesticides here. So this is one. So what I'm showing you here is a sample from 2016. This is one pollen provision sample from Orchard N. And this one sample had 17 pesticides found in it. Of these 17 pesticides, 10 of them were fungicides. Now, that doesn't seem so bad. Like fungicides, how bad could they be for bees? They're fungicide. But over this talk, you'll see like how what the risk is here truly of the fungicides. They also found 3 insecticides. So remember, these pollen provisions were taken during apple bloom. So during apple bloom, apple growers don't spray these insecticides. So these are coming from pre-bloom sprays, or they're coming from somewhere else, right? But they're still contributing to the exposure here. And then we also found some trace amounts of herbicides. So that one pollen sample came from this orchard N here. And so you can see this orchard, there was other provisions that were tested from this orchard, but this orchard showed to be exceeded the US EPA acute exposure level of concern. So, meaning that, and the EPA sets this at 40%. Okay, so back in 2016, when we did this, it was like, ooh, that's not so good. Okay, well, that's just 2 farms. But, Scott and David and Nico continued to do this for 5 more years up until, and up until 2021. And so I just pulled a slide from 2019, a summary slide. And what we typically see is a lot of variation year to year. And so I pulled 2019 because this year, out of those same orchards that had been tested this whole time, we found 9 of those 20 orchards were above, far exceeded the EPA level of concern. So some years it might be 2 farms, some years it might be 50% of the farms here. And in one of these farms, it was over 300% of the level of concern. So risk can change year to year. So what does this mean? So our colleagues have found similar results in Michigan blueberry, New Jersey blueberry, and New York strawberry. And they also find year-to-year variation, and theirs on average varies 30 to 40% of the orchards exceed EPA acute level of concern. So this begs the question, why aren't there requirements to assess real-world exposure after releasing a product? It's just something to think about here. Okay, so now what do we need to know better? Where is exposure coming from? So a colleague of ours, Kelsey Graham, did similar work to our apple work in Michigan blueberry pollination. And she took pollen provision samples from honeybees across 20 different orchards, 20 different blueberry farms, and analyzed the pollen for what they were bringing back as far as pesticides. And so she split it up. She looked at unsprayed farms, organic farms, and conventional farms. And what she found was that on average, all of these farms brought back around 35 pesticides in the pollen provisions. And so this is really interesting because unsprayed farms shouldn't be having any pesticides. They shouldn't be having anything else. Organic shouldn't, they should have other stuff in there, and then we'd expect it on conventional. But she was seeing the same amounts no matter what kind of farm management. And so where are these pesticides coming from? It's definitely like off the farm, right? Or it's from spraying during non-bloom. So Kelsey went out and she investigated further. She went and interviewed all the growers, asked them for spray records, looked at what they sprayed before bloom, during bloom, and after bloom. And what she found was that almost 60% of the pesticides found in the pollen provisions were were pesticides that are not registered on blueberry at any time. So these weren't coming from blueberry anywhere, right? So that's pretty significant. She also found that a small amount was from pesticides sprayed when blueberries were not in bloom. That's predictable. And then also that's predictable because some pesticides linger in the soils and in the tissues of the plants. And then she also found that some of the pesticides were not necessarily from that farm, but they were from they weren't sprayed on that farm, but they were sprayed on farms nearby. And that's not surprising because honeybees like to travel. And then as you would expect, in conventional fields is where she found that there were pesticides that were actually sprayed in there. So where were these other pesticides than ones not registered for blueberry coming from? And could it be these field crops that have seed treatments, or like what Diana talked about, where squash, they have seed treatment as well. So are these coming from other crops? Is it from the wildflowers on the field, along the field margins and the trees? Our our grad student studied a bunch of flowers in trees and found that bees are visiting trees quite a bit. So the conclusion is that many exposures come from outside focal farms. So what does that mean for regulations? You know, how do we deal with that? And also, we found that several pesticides are lingering from pre-bloom sprays. Do pesticide labels need to be changed or updated or more transparent? What do you do with that information? How do we utilize that? Okay, so now fungicides. What's with all the fungicide research these days? Scott McArt, when he was a postdoc, he did this study looking at, it's not the slide here, but he looked at bumblebee declines across the US, and they looked at all kinds of variables. And what they found was that the increase in fungicide use correlated with the decrease in the bumblebee species declines across the US. And so he had always wanted to further ask questions about this. And then Diana D'Souza came, and she was really interested in further exploring this question. And with hers, we start, since he had done that work, these synergisms had started to come in. Like, synergisms are when the fungicide combines with an insecticide and basically makes that insecticide exponentially more toxic. And so she was curious. She's like, is the effects of the fungicide, is that from direct toxicity, or is it these synergisms? So they chose together Captan, because it was the third most used fungicide in the US. And so she had 4 treatments: control, Captan alone, thiamethoxam, and then did Captan plus thiamethoxam to explore the synergy. And to look at realistic levels of exposure and kind of try to understand that level of exposure component, she also broke that down into different exposure levels. And so they picked 100 parts per billion, because in all the pollen samples that come through that pesticide analysis lab, almost all the samples from apples show to have about 100 parts per billion of Captan in them. And then very often they also have 500 parts per billion. So they chose that for the medium, because sometimes there wasn't 500 parts per billion. And then they chose the high level of exposure for Captan to be 2,000, because that's the highest level they ever documented. And they did a similar thing for thiamethoxam. So 10 parts per billion is low, and 70 parts per billion occurs, but it doesn't occur all the time. And then 1,400 parts per billion is the highest level documented as well. And what she found was that Captan increased mortality by 80%. This is a fungicide, and here it's acting more like a beicide, So we don't typically think of fungicides being hurting the insect, hurting our insects. So this was an interesting finding. This was specifically with larvae and pupa. And if you look at this one I just added here, so these mortality rates were similar and not significantly different from the mortality when low amounts of thiamethoxam are applied. So, there again, it's acting like an insecticide. And then the one piece of good news was these last 3 bars down here. This was the combination exploring the synergy. And so this is Captan plus thiamethoxam. And we don't see any synergistic increase in mortality from that. So that's good. That's not happening out in the landscape, and that's good to know. But so she looked at caftan and she thought, but this is the same thing going on in some of our other fungicides, especially some of these ones that are classically known to be synergistic. Like we keep blaming synergies. Is there something else going on? And so she looked at difenoconazole, fluxapyroxide, and fenbuconazole and found similar increases, 63%, 44%, and 52% increase in mortality. And these were not that different from the bioassays that she did on thiamethoxam and cetamiprid as well. There's similar mortality rates. And when you pool all this together, so she pooled low, medium, high for each of these chemicals. She pooled that together. These mortality rates were not significantly different. So that is really interesting. And the most interesting part is that all of these fungicides are, it's a surprise that they're doing as much to bees that we never knew about. So why are honeybee larvae so sensitive to fungicides? We are exploring this more. Scott's starting to do microbiome research in our native bee lab. Brian Danforth is driving some research on native bee microbiome research. So hopefully we can present on that in the next few years. And also I wanted to let you know that EPA is starting to consider larval assays during pesticide registration, but will larval assays be given as much importance as adult assays? And honestly, it seems like they probably need to be given a little bit more, since adult bees aren't the ones typically eating the pollen. I mean, they eat a little bit, but larvae are eating the bulk of the pollen collected. So maybe these assays should be done specifically on larvae. So in collaboration with Dyke and PMEP, we published some pesticide decision-making guides to protect pollinators. We'll be putting out some for small fruits and others for vegetable this spring and summer. And this guide for apple, I think it's a misnomer that we called it a decision-making guide. It should be an education guide, but they're really helpful in teaching you what each of the chemicals has been reported to do to the bees species that they've been tested on. So I encourage any questions and want to let you know that any of you can send in pollen samples to our pesticide analysis lab. Anybody in New York or Vermont or anywhere can. It's $90 a sample. If there's any questions, this is the end of my talk.
Maria, thank you so much. Really fascinating and scary as well.
Yeah, I know. I don't know where. Yeah, we'll see where we go with it and what happens. And
Maria, I had a question. This is Mary.
Oh, hi.
Hi. So you were saying that one of the things that you're thinking with fungicides and why they might be so toxic for bees is the microbiome. But I guess I'm a little bit confused about that, because I think that the LD50s that are created in the lab are usually done with just like a feeding assay that might not even involve pollen. And it looked like even DeSosa's work, was it pollen or, like a sucrose solution?
It was, I believe it was, oh my gosh. I'll have to look at that again. I think it was, you're right. I think it might've been sucrose. It's been a while.
Then in that case, it's like the LD50s might not even be, you know what I mean? Like the lethality of it is not really necessarily tied to the microbiome in that case, if it's not pollen. I don't know. Just
well, so it's still affecting the microbiome if it's being ingested, right? So those fungicides are going in and killing the gut flora of the bee, making them less immune.
Mm-hmm. Okay, that makes sense.
So that's the idea with the microbiome and how how they're affecting it. So
Thank you.
yeah, I think you're right. So that wasn't done by pollen, that was done by nectar. But it would still do the same thing.
Right, yeah, thank you. I had forgotten about
Probably
this.
but like you said, it might be a little bit more, there's somewhat of a wild card there, right?
And Maria, this is Dan Wickstead. If I could ask a question.
Yeah.
You mentioned why aren't there requirements to assess real world exposures after a product is released for use, but there are. One thing, federal law requires pesticides to undergo registration review every 15 years. So
Right.
after it's released, they still have to, they look at relevant literature review to see if they're seeing problems like this. They can also, EPA can require added data call-ins to get more data. And also,
how do they incorporate like those landscape effects
well,
that we were talking about? Like it might be coming from a neighboring farm or from residuals and stuff like that. How do they?
well, they can't with their testing, but that's why they look at the literature to see what's going on out there. And then they will do, if the need arises, they're seeing the LOC being exceeded, they go to semi-field studies. And if that's still showing issues, they will go to full field testing with full colonies to look for effects of the pesticide. The problem does come in where you can't, it's hard to require a test that looks for every possible combination of every possible pesticide that could be out there, because that's infinite. No one can do that. And that's why they review the literature and request further data call-ins if the literature is showing issues.
I'm going to have to jump in here because we need to move on to our next speaker. But thank you so much, Maria. And maybe if you'll stay around and look and chat if any questions do come up.
Yeah, so I'm a graduate student in entomology. As Steve said, I'm in the Thaler lab, and I'm going to talk today about how soil management practices might affect pollinators and crop disease resistance. So the major manipulations that we make to the soil in agriculture, we know can affect above ground ecosystem services. And I'm really interested in how soil management might dovetail with managing both pests and pollinators on farms. And so I'm going to talk a little bit about that generally, and then share some of the research I've done on this topic. And so we know that the nutrients and microbes that are provided by soil shape crop interactions with beneficial and antagonistic symbionts, which can ultimately affect yield on farms. And so it's important to manage soil health in a way that promotes yield while also reducing pest pressures and mitigating negative effects that might occur for beneficial organisms, especially pollinators. And there's some evidence that organic soil management specifically can condition soil in a way that improves crop resistance to herbivory and pathogens, which is interesting because maybe we can apply some of those practices that are used in organic farming to non-organic systems to at least reduce pesticide and fungicide use. And some of the ways that this happen can be by changing soil nutrient profiles, as well as microbes in the soil. And the changes to soil properties that happen from soil conditioning due to some of these management practices can change leaves in a way that is important to pest and pathogen resistance. So leaf nutrient profiles can change due to soil, and so can some of the chemicals that plants produce to defend themselves against pathogens and insect pests. But we also know that the effects of management practices can take time to manifest. So if we implement a certain soil management practice in a single year, we might not actually see a difference in something like pest pressure or yield, or resistance to pests, I mean. But over time, we might see a difference with that management practice. And that's because of this soil conditioning that can happen to microbial community or soil structure, or maybe nutrient composition. So one of the focuses of my research when I think about how soil affects pest pressures is looking at how long-term management decisions over the course of several years can affect pest and pathogen resistance. But I'm also interested in how soil management practices affect pollinators. As I said, There's a lot of discussion about using cover crops for soil fertility and how that might dovetail with providing pollinator habitat. And so if, for example, if we let cover crops flower, we can create an abundant resource pulse of nectar and pollen for bees. And that's like a really great strategy that I'm not going to talk about today, because I'm more interested in how conditioning of the soil could affect the quality of the pollen and nectar that is provided on farm to bees. So I'm kind of focusing on how soil might change the amount or nutritive components of pollen and nectar that bees rely on for food. And so we think that this could happen because soil changes leaf tissue, as I was just talking about, by affecting nutrient uptake and chemical production. And so like, there's no reason to think that that might not happen in flowers as well. And there is some evidence that nutrients and microbes in the soil can change macronutrients in pollen and nectar, amino acids in pollen and nectar, and all of these nutritive traits are important to different stages of the bee life cycle. So Diana and Maria did a really good job of explaining use of food resources across the bee life cycle. So they really need carbohydrates and fats and proteins for all of their activities. When adults are foraging, they need abundant nectar and sugar to fuel their flight and fuel mating, and the pollen is really important for larval development. And I'm really interested in looking at how soil affects pollen and nectar quality in squash for a couple of reasons. First, because it's a common crop that's grown that is affected by disease. So powdery mildew and squash mosaic virus and striped cucumber beetle are all significant pressures, but also because it's visited by these specialist bees that Diana and Maria both referenced in their talks. And so specialist bees have a narrower diet range than what we refer to as generalist bees that forage on lots of different species. And because they only eat pollen and nectar from one or a handful of flowering plant species, any effects that soil might have on the nutrients in pollen and nectar could have an outsized effect on these specialist bees. And that's because they can't really switch to, you know, like foraging on clover or something. And so I'm thinking that soil environment could affect pollen and nectar through a couple of different paths. So in the same way that soil can affect leaf quality, we might imagine that nutrients and microbes in the soil affect nectar and pollen in a direct way, and that could scale up to affect both pollination services and the bees themselves. But we also might imagine that by changing pest and pathogen pressures, that soil could have indirect effects on bee diet quality and bee behavior. So we know that when herbivores chew on leaves or pathogens attack plants, that can constrain their ability to produce flowers or to produce high quality sugary nectar or pollen that bees want to eat. So I was interested in looking at these direct and indirect effects that soil could have on bees. So I've teamed up over the last few years with some folks from Cornell and the Cornell Small Farms program, who have been running some long-term experiments manipulating different fertility inputs in organic plots. And so I collected soil from those plots and also from some fields that were under long-term conventional management on Cornell experimental farm sites. And I planted squash in these soils and then measured herbivory, so insect damage, disease, and pollen and nectar nutrients. And I used summer squash as the model system. And I actually used Success PM that's supposed to be fairly resistant to powdery mildew, which I didn't actually find to be the case in this study. But in any case, I had these organic and conventional soils. I'm not going to talk about it today, but I actually used soils from several different management practices. So within organic, we looked at a few different fertility inputs, and same with conventional. And I'd be really interested to talk to people about that after, just since I can't go into it today. But just jumping to what we found that soil management had no effect on cucumber beetle damage. So regardless of whether plants were grown in conventional or organic soils, they got slammed with cucumber beetle and had equivalent amounts of damage. So that wasn't really what we were expecting to find. But we did find that plants grown in soils with organic management history consistently got less powdery mildew than plants in conventional soils. And we found that across 2 years and in the field, as well as in greenhouse experiments. And then we also saw that plants grown in soils with organic management history had more sugar in their nectar and produced more nectar overall than plants in conventional soils. And they also made more flowers. So on a per plant basis, they're producing more flowers, they're producing more nectar and more sugar per flower. So the carbohydrates that are present in nectar were on a per plant basis, just more abundant in organically grown plants. And interestingly, sometimes the higher nectar rewards in organic plants were due to lower powdery mildew infection. So we saw that powdery mildew also depressed nectar production. So we kind of see a double effect of organic management there. Like it on its own improves nectar production, but also by reducing disease pressure, it can promote nectar production. And then bees visited plants that had more of these nectar resources. But on the other hand, we found that soils grown in organic soils made pollen with less protein than plants grown in conventional soils. And it's a little bit hard to interpret this result because we don't, or actually really either of these results, because we don't actually know what effect these changes in nutrients are doing to the bees. So some of my future work is going to look at that. I have pollen and nectar from plants grown under the different soil management conditions, and I'm going to feed it to bees and see how it affects their larval development. And then from the grower's perspective, of course, the big question is, how does this affect yield? We did see more visits to squash plants grown in organic soils, as well as in soils with plants with less powdery mildew. So the implication of that could be that yield is higher, but it also might not be. So that's something else I'd like to look at in the future. And I think that's all I wanted to talk about today. So I just wanted to thank everyone in my lab and at Cornell Farm Services who helped me with this project, as well as all the funders. And I'd be delighted to take questions or hear some discussion of this work.
Thank you so much, Julie. Are there any questions? I don't see any. So Julie, I do want to thank you and all the speakers for a really fascinating session, a little different than what we typically have at the Expo, and really some things that make us think, and lots of additional research will be coming down the road, I think, from this work. So again, thank you to everyone.
This has been a production of Cornell University, on the web at cornell.edu. This has been a production of Cornell University, on the web at cornell.edu.